Hi everyone, my name is Nasmille Larke-Mejía and I'm a Marie Sklodowska-Curie Actions postdoctoral fellow based at the Volt Center at the University of Copenhagen. My background is in biology and environmental microbiology. I did my 2 BScs and masters in Colombia and my PhD in the UK. I grew up in a small mining community in the north of Colombia, and I would see and feel on the ground the explosion of the earth every day. So this gave me a lot of interest, and I was inspired by learning that organisms can actually transfer DNA from one to the other, known as the horizontal gene transfer, and sort of get superpowers. So this brought my interest into studying microbiology, and I've also always been interested in how can we mitigate the effects of anthropogenic actions like mining. My first project for my bachelor and master's degree was a collaboration that I built between the mining community, the multinational company Cerrejón, and the university where I did my degrees. In that project, I isolated and characterized bacteria that were able to consume the explosive. After finishing my master's degree, I had a small career gap in which I did some teaching to teenagers and chemistry for high school students. And then I came back to academia to do a PhD. I applied for some funding from Ciencias, the Colombian government, and was given the opportunity to travel to the UK to do my PhD. This was where I first was introduced to isoprene. Isoprene is a biogenic volatile organic compound, BVOC, that is naturally produced by animals, microorganisms, and most extensively by plants. It is emitted in small amounts by leaves, but when you think about a tree that has thousands of leaves, and when this tree is part of a huge plantation, the amount of isoprene that is emitted into the atmosphere is much, much higher. It is estimated that about 550 to 700 teragrams of carbon are emitted into the atmosphere in the form of isoprene. And the story doesn't stop there. With climate change, plants are feeling stress because of heat and stress because of other different, for example, drought, and the emission of isoprene by each plant is increasing. So the Center of Volatile Interactions here in Denmark, where I'm based, studies isoprene cycling, and a lot of their work is on isoprene emission. From my PhD, I studied isoprene degrading microbes. So which are these microbes that are helping to mitigate the effects of isoprene emission? I was able to isolate some organisms, do stable isotope probing, some metagenomics with these heavy fractions from the stable isotope probing, and prove that these organisms in nature are able to consume this compound. So the reason why I started my PhD with Professor Colin Merle was primarily to learn the technique, stable isotope probing. So stable isotope probing is a technique that was pioneered by the J.C. Merle lab, in which we can use an isotope of a compound or a labeled version of this compound and follow it from, for example, being in the atmosphere to being consumed by an organism. So we can actually track how this compound goes from being a gas to forming part of the proteins, DNA, and RNA of an organism. And there are 3 derivatives of CYP right now: DNA CYP, in which we study specifically the DNA produced, RNA, and protein sip. We take a microcosm, which could be soil or leaves, we add media and we put it in a crimp glass vial to make sure that the gas doesn't come out, and we add a labeled isotope. If we measure using a machine like a PTR or a GC, we can see the decline in the amount of the volatile in the headspace. That means that there are organisms inside the soil or on top of the leaves that are consuming it. The more of the labeled isotope that we add, the more we are feeding these specialized organisms that are able to consume it. And these are our isoprene degraders. My project is called MicroArctic, and it hopes to study microbial isoprene cycling in the Arctic or in Arctic plants. I'm going to do a couple of trips during my fellowship post in the Arctic, Abisko, Sweden, and I am going to collect plant and soil samples from different shrubs and mosses present in the landscape. My objective with this is to enrich for isoprene degrading bacteria, construct a collection of isoprene degrading bacteria that we can use here in the lab, do some stable isotope probing, and if everything goes well, try and do a mesocosm sip, which would ultimately be a real-time sip on a plant. So why the Arctic? The Arctic is heating up 4 times quicker than any other environment on planet Earth. So with this rapid change, we can also see what will happen to microorganisms and plants present in these environments. As I said before, plants under stress produce more isoprene. So we can see if organisms are going to adapt or if they are going to disappear. I believe that there is power in biodiversity, so hopefully what we will see is organisms that are able to adapt to the changing climate. Thank you all for listening.